So, good evening to all. I welcome you all in this beautiful evening. So, today on this day of 21st February 2021, so I, your host, Dr. Muktinder Singh, so today's, as you are aware that IIHP has been holding these webinars since we started with the lockdown. So IIHP took this initiative to present or continued medical education program. The seniors and our leaders, they have taken this initiative, and IIHP has been successfully organizing these webinars. So today I'd love to introduce you to Dr. Chinta Raveender, who is a microbiologist, who is a homeopath also, and he has been working as former scientist, assistant director homeopathy in Center Council for Research in Homeopathy and Ministry of Ayush. So he is a person with a research background, and his today's webinar topic is on modern emerging concept of homoeogenomics and homoeogenetics, ample of scope for innovative research on cancer. So as we know that at the time of Dr. Hahnemann, the things were different, and today we are living in a different world where the research and innovation and artificial intelligence and genomics and all these sciences and the science conceived by Dr. Hahnemann was at par when these sciences did not existed. Even at that time, the theories given by Dr. Hahnemann in Organ of Medicine and many of our stalwarts who have worked on cancer like Dr. Bird, Dr. Brunet, Dr. Boger, so they have showed the result in it. And if you retrospect those results, those homeopathic medicine selected on the base of similinum has already acted at the genetic level. So this is the beauty of homeopathy, that it is a science of nature and it has that potential to alter those sciences which were even not to show and research in those sciences which were not in existence at that time of Hahnemann. But homeopathy is still relevant to this new age sciences. So I welcome Dr. Chinta Raveender on this platform on behalf of IIHP CC committee and our President of Honor Dr. V.K. Gupta and Dr. M.A. Rao sir. So many seniors are already here. So I welcome all of you to this beautiful evening. So Dr. Raveendra has already shared his screen. So I would invite him to start with his presentation. And in between, you can have your questions and you can type them in chat box, or after his session is over, we'll have an open question answer session. So hope you learn a lot, and I wish Dr. Raveendra that he enlighten us with his insights into the topic and we all get maximum benefit of his talk. So welcome Dr. Raveender. So I, on behalf of IIHP CC committee and this webinar scientific committee, I welcome you to this beautiful evening. So stage is yours, Dr. Raveendra.
Thank you very much.
You can unmute yourself. You can unmute yourself.
Interesting, sir. Good evening to all of you. Respected Padma Shri Dr. Gupta ji, sir. Dr. M.A. Rao, National Vice President, IIHP, and other executive members, and all the participants of this August evening. First of all, let me express my happiness and thanks to the IIHP for giving me this wonderful opportunity of interacting with all of you related to one of an important, as Dr. Muktinder Singh has rightly pointed out, traced back to Hahnemann's writings of 200 years ago. Now, as you all aware about the cancer now, the scope of the disease, how the cancer is emerging, all these things. In my presentation, actually, I'll be talking some of the latest scientific information and with so many modern terms. So don't be very much, do not worry about the big terms like that, because the Hahnemann, which he has invented, the homeopathy with the three cardinal principles, have a lot of relevance to the current modern medical sciences, as well as all the allied thanks and hats off to all the modern medical scientists as well as the allied scientists who have contributed a lot just to speak our own homeopathy, which is invented 200 years ago in their language, spoken long back by our Hahnemann. So I made my presentation in two parts in the sense. First part, I will try to emphasize the scientific temper of homeopathy, which is prevailing now, the cardinal principles of homeopathy a little bit. If you can understand those things, you can wonderfully understand some of the. I will go into in my second part into the basic aspects of the cancer, why the cancer is getting occurring, how homeopathy has relevant to it, and how innovatively out of the invention of the Human Genome Project, I will let you know some of the RNA molecules, how they are wonderfully designed by other allied and modern medical scientists, which is really a lot of booting to all our homeopaths to think innovatively, to ponder many times, and ultimately to have a wonderful scope on cancer. So with this background, so this, because the beauty of the homeopathy is, thanks to all the scientists, whatever they have invented, which is really contributing to something about homeopathy, saying that homeopathy is not at all a placebo and it is much more advanced science, which we can speak not at the level of cellular only, cellular and molecular biology, and we can talk in terms of sequence. 25,000 of the genes language can be expressed. Homeopathy can 25,000 language of the genes. So with that background, I will just share initially about the scientificity of the homeopathy. It got related to some sort of ultra high dilution and our personalized concept. Like, can I go to the second slide? So just to overview the total scenario and status globally, you all know characteristics of the people taking homeopathic treatment is all over the globe. So, number of clinical trials with standard protocols are being undertaken, including lots of RCTs today, including our CCRH also. And there is a lot of fundamental research in homeopathy is being undertaken in collaboration with different allied sciences. We have a experiments physics related. We have a biological models, that is animal and plant models, microbiology related in vitro and in vivo studies, and we have a genomic, proteomic, signalomic pathways research, electrophysiological parameters homeopathy has been utilizing, and various biomarkers, etc., including lots of cell line studies. So, basic research outcome of the modern medical as well as the allied sciences, I'm going to share in upcoming slides, which is supporting for homeopathy for improving the success rate in our clinical applications. This is our prime duty to look actively into the research of the other scientists and make it use for therapeutic application, thereby increasing a lot of homeopathy scope in the clinical studies, which is more useful for all the practice. Is there any problem? So now let me share a little bit about the basic principles of homeopathy. As you are all of you know, we have a law of similia, then next law of individualization, and law of minimum dose. So these three principles, how wonderfully all the modern medical scientists, as well as again I will say the allied scientists, have contributed wonderfully a lot to explain these three basic principles. So in my couple of slides, I will try to see what is the law of similia and what is its significance in the modern biological similitude principle, which any microbiologist, any cellular and molecular biologist can speak of the biological similitude principle, which has a lot of similimum to our law of similia invented 200 years ago by our master Hahnemann. So these law of similia is nothing but in the modern concept, we will experience this in concept of vaccination. Everybody knows, I did not explain it. And ultra low dose concept. This is something very new concept that also supports law of similia in homeopathy, which I will share in due course, my slides. Then the modern concept of immunosuppression, immunomodulation, immunostimulation, and including modern immunotherapies have a lot of relevance of law of similia. Then certain basic operative mechanisms which are of very immunological important in immunological functions, like immunological surveillance. I'm not talking in a big way, I'm putting in a simplest language. Immunological surveillance is a wonderful immunology mechanism which is basically intended for the prevention of cancer. So if one understands this, we can wonderfully say that how can we approach for cancer. Another important function of immunity is self-tolerance, which is basically intended for the prevention of autoimmune disorders. And the other hypersensitivity related mechanisms of our immune system are for the prevention of allergies and other immune competent functions, or to prevent the immune deficiency disorder. And one of the important thing is, we all think of now how we will be healthy. If our all cellular DNA should get repaired, then only we will be healthy. See, even this law of similia exists. See, there is a mechanism, constant damage of cellular DNA, and there are the different types of repair mechanisms. If you very deeply look into this repair mechanisms, see, there is again the law of similia we can wonderfully experience. Next. So this law of similimum, it exists several levels. So for example, competitive and non-competitive inhibitory mechanisms. There are certain physiological mechanisms in our body have a law of similimum. They are also law of similimum. So with this, and there are structural analogues which are similar, which have the capacity to inhibit the metabolic systems. See, there is an important similar function in this component also. Antigen antibody reactions, you all know. There is one etiological similimum. See, excretion of toxin, arsenic from the experimental animals by means of a potentized arsenic. See, we got a wonderful public health project in West Bengal. It is out of Dr. Bakshu's genetic research. So in a similar passage, see, etiological similimum, the toxin can be relieved by the potentized toxin, so which we are planning actually at Udanam Nephropathy. A wonderful concept where even Government of India or in the Ministry of Health can go in a big way by based on this principle for the treatment of CKD patients of Udanam region. That, if time permits, I will share that also. So in this particular way, the semi operated at different levels. Thereby, we can widen the scope and application of law of similimum. So Hahnemann has wonderfully given us a basic truth, which we can widen with further research. Next. So now I have shared something about our Hahnemann's law of similia. Now, one of the another pillar of homeopathy is law of individualization. So this has a lot of relevance to the coming or existing or almost yet to come in few of the countries, that is personalized prescription based on the Human Genome Project. So this is going to influence in a big way globally, even including the prescription allopathy. So then let us share some relevance of our law of individualization to this personalized prescription proposed by the modern doctor. So let me share a little bit about the genes so that you can wonderfully appreciate everything about homeopathy. The Human Genome Project, that is nothing but sequencing of human DNA containing 25,000 of the genes in a single cell. So this already mapped out by means of contribution of various countries or various centers over a period of few years. So little bit to state about, see, every cell has got an housekeeping gene. They are expressed in every cell for maintaining the cellular viability, that is life and death of the cell. So there are other genes which are expressed only in some cells, like that in liver cell, heart cell, etc., depending upon the function it has to perform. So if we take this into context of homeopathy, for making an optimum biological or psychological functioning is nothing but health of an individual, different types of genes must be expressed at a correct level and at a right time. So expression of wrong genes at any time or overexpression or underexpression of some of the genes brings on various diseases, including cancer, by the activation of a cell cycle at a wrong time. This is which have relevance to our today's topic. Next. So, how these 25,000 of the genes are influenced? Hats off to Dr. Hahnemann, how he's looked into. See, many internal and external factors, including food components, thought process, radiation, exercise, sound, lack of sleep, exposure to the environmental toxins, continually disturb the optimum transcriptome expression. See, this transcriptome is nothing but expression of a set of genes at one particular time which brings out a character. So that is nothing but the transcriptome. So to maintain optimum health, the body is continually striving to maintain the right level of expression of thousands of genes in each cell. See, this is nothing but Hahnemann has spoken in the language of vital signs. So now we have to speak in terms of genes. So in this context, practice of homeopathy can be considered as a way of promoting the health by helping the body to maintain the optimum expression of each cell's transcriptome. See, these trillions of cells should have a harmonious expression or harmonious up and up regulation, down regulation of the genes. Then only we will enjoy a positive health. So just based on this background, the sequence-specific homeopathic DNA preparation also have come. Macroimmunotherapy and micro-DNA therapy. I will talk later on.
वो पेरीमीटर वाला का चैप्टर ला।
अरे छोड़ नहीं, तो ग्रुप के ऊपर तो पूछ।
नहीं अभी नहीं पूछेंगे अभी।
अभी क्यों नहीं?
तो मुझे ये कर, हम पेरीमीटर के चैप्टर में
were found to promote and support the health and correct the disease and promote the natural super health. So microDNA therapy molecules have been designed for targeting the health promoting genes. Just I will little bit show something about other genes other than cancer genes. Here there is one gene like KL17 whose activity declines the age, and other genes that promote the natural super health. So here actually we can target a specific gene with our ultra highly diluted DNA molecule. So the natural super health, also known as the heterosis, occurs in all the organisms. It is the highest level of health that can be achieved naturally, otherwise known as refining our own DNA for better health. The advanced homeopathic DNA gene targeting molecules and are very safe. For example, MTHFR gene product is associated with homocysteine. You all know the risk factor of homocysteine. One of the risk factors for many diseases. So there is an ample of evidence that highly diluted homeopathic medicine should be able to alter the gene expression and act at genetic level. And to say something about the Luc Montagnier, everybody knows, the inventor of HIV virus, correlated the homeopathic principle of dynamization in emitting of the electromagnetic signals from the aqueous nanostructures derived from the bacterial DNA sequence. So, in this background, London is the first country for implementing the human genome data clinically. Hope this will be done by so many other countries. So now I will come to our minimum dose. I have covered law of similia. I have covered about little bit scientificity of the personalized approach. And now let us look at wonderful another pillar of homeopathy, the law of minimum dose and modern ultra low dose concept and nanotechnology also, another science which is contributing to our ultra high density. Now I will only share some of the important things just definitely they will provoke our thought process to invent more drugs in homeopathy in the light of different diseases occurring in this modern era. So, for example, growth stimulating agents, pesticides, neuroimmunomodulators, anti-tumor drugs, anti-metastatic, antioxidant, antibiotic, radioprotectors, we all know. So when these are all experimented in ultra-low doses, they brought the favorable changes at bioobjective sensitivity at cellular and molecular level and at the organism level, which has a wonderful relevance in the treatment of so many diseases. They brought the favorable changes in the molecular organization of the biostructures, particularly the cell membrane. Whatever we speak of our drug or allopathic drug or Ayurvedic drug, it has to work through the cell membrane only and it has to go to the nucleus. So there are certain four-ball esters or the substances, these are the substances, have the ability of modulating the various important regulatory cellular mechanisms. So there is one lipid peroxidase system. E is connected with the cell proliferation
perimeter ka chapter
and growth.
I will have to give you something from it.
Which I spoke, these elements or molecules, whatever it may be, there is TPA and DDP. These are the two four-ball esters. In ultra low dose, would be able to inhibit the lipid peroxidation system at 10 to the power of minus to the 10 to the power of minus 16, with a maximum effect at 10 to the power of minus 12 to 14. See, this is how wonderfully they have highlights the efficacy of different potencies. So then coming to the antioxidants, everybody knows this action. They usually regulate the physicochemical states at lipids of the cell membrane. So these antioxidants, the so-called antioxidants which you purchase from the medical stores, when they are experimented in ultra low dose, is nothing but highly diluted form of these antioxidants at the power of 10 to the power of minus 30 in concentration, produce cooperative modifications in the cell membrane. The newly formed state is more favorable and stable. So another small important, everybody knows vitamin E. Its action, you know it. Then when this vitamin E is diluted and experimented, it could able to inhibit the phosphokinase creatinine, C-phosphokinase activity in human sarcoma cells. Vitamin E, ultra low doses at the 10 to the power of minus 19 has brought more favorable changes. So this all shows how best we can borrow a lot of pharmacology from the modern drug. So there are some of the limitations. The ultra low dose of endothelium, which is a protein, endothelial protein, showed the favorable effect on coronary resistance of the blood flow, having scope in CAD. And another ultra low dose of the neuropeptides, everybody makes neuropeptides, showed a favorable adaptive mechanisms in our body. Then ultra low dose effects of an opial alkaloid, BALZ, is an opial alkaloid, inhibited the respiratory process, showing its efficacy in respiratory allergies. Everybody knows the thyroid stimulating hormone. Its action, everybody knows it, including modern doctors. But when this TSH is experimented in ultra low doses, it has decreased the conversion threshold. Can be experimented clinically. See, everybody TSH thinks it can be associated with only thyroid. But when it is diluted and experimented, it is able to show its efficacy in conversion. See how the principle is like homeopathy. Next. And everybody knows this methotrexate, an anti-immunosuppressive drug, anti-tumor drug, like which they are giving in cancers as well as in psoriasis and rheumatoid arthritis as an anti-cell divider. So in physiological dose, is an immune suppressant, and in ultra low dose is an immunostimulator. Can be experimented clinically in rheumatoid arthritis and psoriasis. So similar favorable results were observed in ultra low dosage experiments of histamine on basophils, that is basophil degranulation. This acetyl salicylic acid, aspirin C, in ultra low dose, it has an efficacy on intravascular coagulation. So finally, coming to another tumor necrosis factor, adriamycin, cisplatin, these are all anti-cancerous drugs. When they are experimented in a cumulative and synergistic action, when they are, see, this supports the combination of similar drugs in higher potencies. See, they were saying similar drugs, when even we also know our homeopathic drugs are not single drugs when they are experimented in ultra low dose, like lycopodium consists of 56 different types of nanoparticles. And finally, there is prazosin, which brought a lot of favorable changes in alpha loaders. So this is contributing a lot to our law of minimum dose. Next. So now, everybody knows about this homeopathy and nanotechnology. Let me also share one of the slides. So then clinical application also is, everybody knows the outcome of this IIT Bombay and IIT Delhi and CCRH also. So the recent outcome of the nanotechnology says that a revolution is going to happen in every field, like pharmacology, textile industry, construction industry, agro-veterinary industry, aircraft engineering, etc. So thereby influencing the human life in a positive way. So the materials prepared out of nanotechnology is going to have a least expenditure, is similar to homeopathy, greater durability of the product, and is more eco-friendly, thus benefiting the whole mankind. So till 2010, the way you are speaking today, in after 10 years again, after a decade, there was a dilemma about the ingredients of ultra-high dilution of homeopathic medicine. And recent experiments at IIT Delhi and IIT Mumbai have demonstrated the nanoparticles and nanopictures in homeopathic medicine. The recent advances in modern pharmacology, nanopharmacology, nano drug delivery systems have a relevance with homeopathic ultra-high dilution. So there was a presentation in the recent nanotechnology conference at Hyderabad with a mention that gold in the nanoparticle is one of the best medicine for cancer. So doesn't have a relevance with the homeopathic medicine of aurum metallicum? Next. So now, see, in this background, hats off to Indian scientists. India has also contributed a lot. I think everybody, when they look into, they will understand that the whole RNA molecules which were being developed by the international patents which they got are out of the research by our Indian scientists, none other than Dr. Khudabakshu. So let me share his work, because his work has the basis for the new molecules which people are experimenting in our homeopathy, which I am going to share. See, homeopathy has been confronted with certain major issues, such as everybody knows. Dr. Khudabakshu has contributed a lot for this point. See, how is the medicinal property of the homeopathic drug transferred to and retained by the vehicle? This is a very big question. How can it act in a biological system in the absence of any original drug substance with ultra high diluted remedies? The plausible mechanism and pathways of the biological action, including mysteries of like cured by like. It was demonstrated through the controlled studies the ability of the ultra highly diluted homeopathic drugs in rendering the protection or repair of the cytogenetic damages inflicted by the whole body X-ray irradiation in a mammalian model, mice in vivo. So they have examined, this Khudabakshu's team has examined the cytogenetic damages in arsenic, cadmium, mercury, and stannum intoxicated mice, and their remediation by the homeopathic drugs derived from the agitated ultra-high dilution of their respective salts, deploying many scientific protocols. See, his genetic research has a lot of background to the new invention of the molecule. Next. So they have conducted a human trial on efficacy of arsenicum album against the groundwater arsenic contaminated. They showed a way also. Now the global scientific community is looking at the Uddhana homeopathy, but homeopaths are not coming forward to have a similar experiment, which we have a lot of scope. So it was tested, efficacy of some of the homeopathic remedies in cancer of the mice and thalassemia in humans. They have produced the nano capsules of the homeopathic mother tincture and their bioactive components, and induced the nano precipitation of the silver from the silver nitrate by the homeopathic mother tincture, and characterized them for their physicochemical properties and biological action. So they have also studied the tissue distribution of these nanoparticles, precise mechanism and pathways of their action that involve in certain signal proteins and their pathways, both in vivo and in vitro. So this approach made a significant contribution towards elucidating the role of drug nanoparticles in inducing the memory of the water and mechanism and pathways of the action of homeopathic remedies through the epigenetic modifications. This is something beyond genetics that supported the gene regulatory hypothesis of homeopathic drug action. So this basic work became the foundation for the bringing of the sequence-specific DNA or RNA molecules into the homeopathy with international patents, which I am going to share in future slides. And now coming to, so with this little background of the great research done by our colleague scientists of medical, brother like Alopus, and as well as all other scientists, now we can simply contribute to, we can bring these two concepts into the homeopathy, homoeogenomics and homoeogenetics. So let me share something about what is homoeogenomics. The homoeogenomics refers to the studies that focus on understanding more about the interactions between the homeopathic remedies and the genetic blueprint of the genome, nothing but an individual constitution. So this is homoeogenomics. Such studies involve identification of which gene or the genes are targeted by a particular remedy. In our drug proving, we are only making down the symptoms. The same drug proving we can speak in the language of human genetics also. Future, we can take a lot of studies. The whole homeopathic drug proving can be spoken in the language of genetics. So now, homoeogenomic studies also refers to the investigation to understand the epigenetic modifications that are associated with rearrangement of the expressed target of gene. So now coming to the homoeogenetics, this refers to the studies which make us to understand the different inherited forms of the genes interact with the remedy. Nothing but how an individual constitution acts towards a particular remedy is homoeogenetics. Homoeogenomics is how the homeopathic drugs can influence the blueprint of our genetics. Now I hope it is a little bit clear to all of us. Next. So now, again, to overview on this slide, Hahnemannian homeopathic concept and modern concept. Law of stimulum, biological similitude, we have looked into. Law of individualization and modern application of bioinformatics from human genome project, we have looked into. Law of minimum dose, ultra low dose concept, we have looked into. Homeopathic ultra high dilutions and nanotechnology, nanopharmacology, we have looked into it. One of the another important pillar, supporting pillar is holistic concept of homeopathy is wonderfully explained by the systems biology. Modern systems biology is nothing but supports holistic concept at cellular level. We are holistic concept, we are talking at an organism individual level. Systems biology speaks the holistic concept at a cellular level. See how beautifully correlated with this. So you know the concept of miasms, flora, psychosis, syphilis, concept of modern pathological defense mechanisms, acute inflammation, chronic inflammation, nothing but granuloma formation, autoamputation or cavity formation. We have it. Our all miasms can be, classical miasms can be wonderfully spoken in the language of these pathological concepts. Next. So now, see, that is the background. This is how Hahnemann came. Hahnemann's directive and the basic objective of homeopathy, everybody knows it. First aphorism. The physician's high and only mission is to restore the sick to health. The results of the Human Genome Project and the new properties of DNA have now been used to bring out a new remedy. See, to support Hahnemann's first aphorism, we can research further, we can bring a lot of wonderful drugs into our science to fulfill our Hahnemann's first directive, the physician's high and only mission to restore the sick to health. Next. So now, a little bit about clinical application of these things. Generally, homeopathic remedies do not contain any pharmacologically active substance, though nanoparticles are there, and are found to have a nanoparticle of the original medicinal source. The transcriptome, which I've already showed, refers to the set of genes that are expressed in any particular cell or a tissue at a particular time. So that's why our behavior, our disease characters are nothing but expression of a set of genes, that is nothing but a transcriptome. So homeopathic remedies will be able to alter the health characteristics of an individual by rearranging the transcriptome, that is rearranging the expression of various genes that affects those particular health benefits. Next. See, almost 2 decades ago, the idea that the homeopathic remedies could interact with the transcriptome and influence the expression of various genes began to be formally addressed by Professor Gudivada since 1997, which I have already shared. His contribution for the last 2 decades has wonderfully made a lot of scientists to work on patents, which I am going to share. So since then, many studies have confirmed that homeopathic remedies do have the capacity to increase the expression of many genes. This I will share in a couple of slides. Next. So now, little bit share with me the classical drugs, how they are acting on the scientific investigations have discovered that individual remedies such as Condurango, Hydrastis tendencies have the capacity to rearrange the expression of many genes, well over 100 genes. Gelsemium semipervirens has the ability of downregulate the expression of 49 genes and upregulate the expression of 6 genes in the human neuroblastoma derived cells. This is only to appraise you how we have proved that our deletions can act. This Phytoleuca decandra will promote the expression of EP53 and BAX genes which are associated with cancer. So I will share. Each of these remedies rearranges the expression of a range of quite different genes. So therefore, with a view to advancing homeopathy, it became an important to develop a way to support health promoting genes exclusively. How? See, from our provings, we are now understanding. Now, when you understand the whole entire human genome, we can definitely specifically try to target the different genes which are the basic cause for various diseases. Next. Then how to target this specific gene? This became possible due to the pioneering work of a homeopath, Dr. Jenner, many of must be knowing, in the late 60s, more than 5 decades, 6 decades ago. As a part of his seminal work, he confirmed that potentized DNA molecules with a defined nucleotide sequence would be used to target and modulate the activity of genes that were involved in fighting the infection. See, initially they've looked into the infection, now we can look at the cancer. Next. So, just to speak something about DNA, in homeopathic DNA, see, we have in homeopathic DNA also. DNA has long been used to prepare homeopathic remedies. It is included in Materia Medicas, compiled by the practitioners such as O.A. Julian, Dr. Jenner, and others. So it is also included in the various homeopathic pharmacopoeias, like United Pharmacopoeia of United US Homeopathy Pharmacopoeia, and it is chemically isolated from either sperm of the fish or from the thymus of a cattle. See, which we are classically purchasing in homeopathic DNA is this DNA, not a sequence specific. So it is of unknown sequence. We don't know the sequence and purity, and it also we do not know, and it may contain the whole DNA also. Next. So various homeopathic DNA remedy preparations have been subjected to the proving process by a number of practitioners like Jenner, Julian, Robin, and others. So because a wide range of symptoms of ill health are induced by the administration of homeopathic DNA preparation, as a part of the proving process, it follows that homeopathic DNA has the capacity to target many genes that synthesize the proteins which promote a wide range of disease symptoms. So this is a modern explanation for our drug. Next. So homeopathic DNA, on other hand, following administration of homeopathic DNA, many health benefits have been recorded by the practitioners like Dr. Suvarna. So these findings confirm that homeopathic DNA can also target the genes that encode the proteins that resolve many symptoms associated with the different forms of ill health. So the use of DNA molecules with a precise nucleotide sequence to prepare the legitimate homeopathic remedies have been validated by the Medicines and Healthcare Products Regulatory Agency. This MHRA is nothing like, it's like an FDA of America in the Europe. So because on this, based on this work, they have granted patents also. So these patents we cannot underestimate. Next. So now See, when we have a DNA in our homeopathy, let us understand some of the properties of DNA, which again helps us to use homeopathy DNA in a better way. The sequence-specific DNA molecules are potentized and administered in a solution to take the advantage of some of the recently discovered new properties of DNA. So, in a solution, they can communicate with and be attracted to another DNA molecule with the same sequence. See, this is something drug action we could able to say. When mechanically stimulated and in solution, they can also emit the sequence-specific electromagnetic signal that can be recognized by the DNA molecule with the same sequence. See, this is pointed out by Dr. Luke Montigner, supporting homeopathy. So, this DNA-mediated signaling cannot be measured when the DNA solutions are diluted more than 1 in 10 to the power of 12. So, that is nothing but 60 potency. Beyond 60 potency, we cannot measure. So, that's why they are being prepared in 60 potency. So, that is why these remedies are prepared in 6 potency. The gene activity can be readily upregulated by the amplification of electromagnetic signaling. There is already proof. If we can produce electromagnetic signal, it could be able to regulate the genes in a desired format, either through upregulation or to the downregulation. And now here I will share a little bit only about the sequence-specific molecules. Then straight away I will go to the cancer, because after DNA there is a lot of limitations for this DNA also, and those scientists enter into the era of RNA molecules. Just before that, so all these DNA molecules that are used to prepare in these molecules are in the double stranded form. So they are in the order of 400 base pairs in length. So these are manufactured by an international big genetic company, GenScript USA, using the classical DNA synthesizing technology, the polymerase PCR reaction, polymerase chain reaction. So they are of precise sequence, length, concentration, so that remedy preparations are standardized and completely reproducible. So one cannot escape if it can be checked in other labs. So they are guaranteed risk-free because they do not contain any DNA sequences of the foreign or virus. See, that's why now many our homeopaths can come across now. Our drugs produce cancer, then how can you treat that? Then they said when we speak about the genes, they immediately question if your drug triggers a wrong gene, then you develop RA, do you develop cancer like that. But the sequence which we have identified is a safer sequence. So that we can explain with so many scientific publications. So the sequence of each of these molecules is determined by the reference to a sequence of the targeted genes reported as a consequence of the Human Genome Project. So the lot of information is available for a particular sequence, and it can be reproduced in other scientific reports also. Next. So these remedies are developed to address a particular set of genes whose genes expression we want to have a suboptimal or more or less like that we want to regulate. The symptom pictures generated by the reduced expression of a particular gene have been accurately recorded and confirmed by the many years of world-class scientific research. See here, out of Human Genome Project, we are studying proving symptoms in a reverse way. Before the disease, we are understanding the proving. So later on, we are targeting it. See how wonderfully we can make use of this entire Human Genome Project. Next. So there are a number of reasons why the expression of a gene may be suboptimal. See, when we should prescribe these drugs also, let me share. For example, the expression of some of the genes declines with the age. Most important health promoting anti-aging gene is discovered is AL, is one of them. So thus, the people when they are aging, everyone would try to get benefit of protecting their KL gene. If the KL gene is protected, they enjoy longevity, slow down of the aging process. We can target that KL gene with a similar sequence-specific molecule. This is like this. We have we can touch up so many solutions. Yeah. Next. So on the other hand, some of the remedies are only required on a need basis. For example, if we were looking at corona, which targets the IL-10 gene in the case of allergic asthma. Like that, we can target these infection protecting genes also, immune protecting genes also. For more information, see, there is a the website here. You can just go, but I will talk something beyond that also. Next. So these I am not going to do. These are some of the different genes which are just we are correlating, which we are going to have a lot of clinical applications. There are some 46 genes which are already patented. They're already available on the net. You can go through and you can read their Mater Medica, and if you want, you can make use of them also. If anybody wants to take research on these drugs also, they can take lots of clinical trials also by adopting all the clinical trial registry procedures. So just only to look at ST6, KLO, IL7, like these are the genes. So in a similar fashion, another paragraph, and in similar fashion, like others are also. So now let me share only these 5 sequence-specific DNA remedies which are targeting cancer. So, for example, TP53, OX40, TP53 is meant for the apoptosis or anti-angiogenic mechanism for prevention of cancer. OX40 is to kill the cancer cell. This KEAP and TAP is for tumor suppression, anti-metastatic. IL7 is for repairing our immune system. This RBM4 is which can be used in all types of cancer. PRC is DNA repair and tumor suppression. And of course, these are available. But out of our experimentation, we gave a lot of feedback to the the original patent scientists. We have showed this. No, this is not that much of improvement in the cancer cases. We could able to almost prescribe around 25 lakhs of prescriptions for advanced stage of cancer. We could able to identify some of the outcome, but that's all we have shared with them. Then these patents, they have already keeping these DNA remedies in suspension, and they are coming with a RNA molecule. That little bit I will share in my future slide. Next. See, now we have learned something about basic research of homeopathy and other sciences. Now, actually coming into the cancer. So, if we wanted to treat the cancer with our classical drugs or with this new innovative drugs, we should know a little bit about cancer at a deeper level. So that I will not go very much deep, but I will share. So in the recent times, now we will talk something about cancer. Scientists have made an important new discoveries about the biology and treatment of cancer, so which applies to us also. So development and progression of cancer is largely due to the increased genetic instability of certain genes in the precursor cells, that is cancer stem cells. So it is now clear that development of the cancer is a multi-stage process. Everybody knows it has not developed over months or few months or few years. It could have been developed over decade also. The genetic instability or the alterations in some of the structures of DNA plays a pivotal role in the multi-stage process of the development of cancer, and development as well as progression of the cancer. So now, see, when we look at the basic, the multi-stage process, what does it include? Which contributes to the development and progression of the cancer includes, there should be maintenance of signals that promote the continued cellular proliferation. Is the basic need of the cancer. Promotion of the uncontrolled replication of the neoplastic cells. Inability to respond to the messages from the growth suppressor. See, suppressor genes, when they are arresting cancer, they do not respond, then cancer increases. Resistance to the mechanisms that instruct the damaged cells to commit the suicide. See, everybody knows, apoptosis is the natural killing of the cell itself, programmed cell death, which is hampered. Again, there is a problem. Enhance the ability to form a tumor-specific blood supply. See, cancer tumors, they have their own blood supply. Activation of invasive properties of the neoplastics. You all know it, how they are very much invasive property. Ability of the neoplastic cells to avoid the mechanisms of immunity. See, though we have an immunological killer T-cell mechanism, that also fails in cancer. So development of the new metabolic pathways that provide the cell. See, cancer patients, though they do not eat, but the cell grows it. So With the process of metabolic pathway, it tries to utilize lot of glucose from our body for its growth. So when these parameters are there in our body, cancer can be unchecked. It grows like that. Next. So now a little bit about homeopathy. It was postulated that homeopathic remedies could deliver their benefits by interacting with the genetic blueprint. The results of many homeogenomic gene expression studies have confirmed this postulate, again by Dr. Gudivada. The results of the homeogenomic studies have started recognizing which of the estimated 25,000 human genes are targeted by the different homeopathic remedies, and the expression profiles of these targeted genes are rearranged. Homeogenomic studies have also shown that homeopathic remedies can also facilitate the epigenetic modification, that is DNA methylation. DNA methylation plays an important role in the control of expression of many genes related to cancer also, which has significance in cancer. Next. Understanding of the genes targeted by the different homeopathic remedies taken together with the information about the function of the proteins encoded by the target genes provides an ample of scope for the research in the management of cancer, which I am going to share. So the homeogenomic studies support the applicability of the frequently used homeopathic remedies in the patients suffering from cancer, particularly with respect to the upregulation of the gene TP53. Next. So now, homeogenomics refers to studies that focus on understanding more about interactions between homeopathic remedies and the genetic blueprint, as I already shared you. Again, the homeogenetics is nothing but how the individual is reacting. Now, let us come specifically to this cancer gene, particularly this gene TP53, which encodes for a protein known as P53. This P53 is one of the most biologically important proteins. The whole cancer research is revolving around P53 gene only, molecule that plays an important role in many critical biological events in the development and progression of cancer. Next. So, I will little bit share more about this. The damage to the DNA, particularly to the genes that control the cell cycle, is one of the important etiological factors in the cancer. The P53 works in a different ways to resolve the cancer. It plays a role in apoptosis, that is programmed cell death, genomic instability, nothing but DNA repair, it is an important role, and inhibition of angiogenesis, which to be hindered in cancer, that is prohibiting the formation of blood supply to the tumor, and inhibition of the activity of an enzyme called telomerase in cancer cells. See, that is the importance of this P53. In 4 ways, it tries to protect us from cancer. When the things go out of order, when P53 gene goes out of order, people will get cancer. Next. Now, little bit P53 and homeopathic studies I will share to get a lot of thought-provoking ideas to the participants and many. The role and significance of the diminished P53 activity in the development and progression of the different types of cancer. Many homeogenomic studies have focused on identification of the remedies that support the expression of P53 gene in many in vitro and in vivo studies. The research work done by the various medical allied scientists have paved a wonderful way for improving the success rate of homeopathic prescriptions with a new innovative DNA and RNA molecular drugs with a special emphasis in the management of cancer, as well as certain burning issues like multi-drug resistance of the cancer. See, we are not only having in secondary cancer, we have an answer to certain burning modern, these we can say that the nightmares to the modern oncologist, modern radiologist, modern radiation therapist, like multi-drug resistance. How best we can address this also? Metastasis, one of the big nightmare of modern oncologists. There is scope of homeopathy. Angiogenesis, another nightmare, etc., are associated with the modern chemo and radiotherapy. The new approach is based on the strong scientific footing, throw a lot of light for conducting the research in cancer with a new innovative development of drugs in homeopathy, particularly DNA, keeping DNA away, and we are talking about only RNA molecule. Next. So there were already international patents on these DNA and RNA molecules, because this gives a lot of scope for directly taking to the research by any of the organizations. Molecules in homeopathic preparations and potency. So the utility of the ultra-diluted RNA molecule as a means of reducing the activity of the specific genes has been confirmed in a grant of patent, which is now expired. See, if we may directly look into, by hypothesis, we may do a lot of research, but we can directly look into the patents and directly design a lot of experiments based on patents also. Even including certain expired patents are open a book for anybody to take them and reproduce them. Nobody will deny it. The patent will be there for a particular period only. So now these DNA and RNA patents have already expired. If with the help of modern genetics, modern oncologists, we can look at these expired patents and find out lot of new and innovative pathways with the newer and newer molecules. That's my aim of objective of this particular presentation. Anybody can think in any way for bringing any drugs into the cancer management. Next. So now I'm coming to the final part of my presentation. I'm directly coming to this RNA molecule. The 2 decades ago, homeopaths Dr. Jenner and Marital developed a remedial system using the ultra-high diluted gene-specific RNA molecule. This system, which uses the ultra-highly diluted potentized RNA molecules with a precise sequences, was clinically proven and validated by this MHRA in the UK. The significance and validity of this technology was realized by the scientific interrogative community because it was granted a patent. The details are available at that particular expired patent. So this now expired ultra dilution technology has been used to prepare a series of remedies designed to suppress the activity of the genes that have been shown to promote the cancer progression and reduce the effectiveness of conventional anti-cancer therapies such as the chemotherapy, radiation therapy, as outlined previously in the course. Next, please. Now, if any homeopathic questions now, do you have a treatment for cancer? We have an answer. Very little of yes, and most of the times no. We are not that much there enough to speak that we have a treatment for cancer. Not only for cancer, even including, though there is a lot of scientific evidence is also there, still we are unable to provide a wonderful solutions to the biggest problems like lifestyle disorders or autoimmune disorders like that, as per to the demanding by our bureaucracy. But still, if we can little bit wonderfully, aggressively go into the research in a collective way, definitely we are going to show a lot of answers to the bureaucrats for the existing illnesses. Had we had a wonderful drugs for so many skin diseases, nowadays the dermatology clinics would have not been that much. Had we had a lot of answers to the cancer, such a the existing cancer hospitals are loaded with thousands of patients. So now, sir, so that's why now, if we can look at this, not only we have this, of course, this is to be experimented a lot with a lot of scientific protocol, but there is a lot of ray of big hope, not only in checking the cancer, and even we can have some of the answers to the problems of this modern chemotherapy, nothing but multi-drug resistance, nothing but metastasis, that I will share in this. I am specifically going into these particular molecules and their clinical applications. Now, many of you have been treating a lot of cancer cases, but you can look into these things and you can also get a lot of information out of it. You will become curious to use these drugs also. So for that purpose, now we have just named some of the names like that. This Onco-Resicare. This is RNA molecule of ABCB1. I will not very deeply go into it. I have provided a website. You can wonderfully go through it, but I have made in a simplest language. When it is used, how it is used, how to use it, I will share with my colleague homeopathic. So this remedy should be administered to all the patients who receive chemotherapy. See, why? The gene MDR1, multi-drug resistant gene, also known as ABCB1, encodes a membrane-bound protein that protects the cells from damage by the toxic substance. See how beautifully It does this by expelling the toxin from the cell before they can cause the damage. Here I want to say our own cell has got this particular gene. If any foreign material enters into cell, it tries to throw it away. See, that is the character they have identified out of Human Genome Project. See, all these modern chemotherapy drugs are taken as the foreign drugs to the cell, and they will see when they are trying to treat a cancer cell, the cancer cell, the multi-drug resistant gene is overexpressed and they're throwing it away. See, the so-called modern immunotherapy drug or modern chemotherapy drug, out of their over hundreds of years of over a longer period of research, simply it is throwing away. See, that's why many drugs, including the chemotherapy drugs, are recognized as a toxic substance. Therefore, they are expelled from the cells in which the ABCD1 gene is expressed. See, if a cancer patient, if the treating cancer patient with a particular chemotherapy drug found to have this ABCD1 gene is overexpressed, the whole chemotherapy drug goes in a zero way, rather it gives a lot of side effects. Next. So unfortunately, many cancer cells express this gene. See, ultimately, again, this is told by the medical oncologist only. Every cancer cell is overexpressed with this gene, saying that the so-called chemotherapy drug is not going to work. So in such cases, because chemotherapy drugs are recognized as a toxic, they are shunted out of the cancerous cells before they can be used as any medicine. How pity. They are trying for treating, but it is throwing away. So this is a major clinical problem for chemotherapeutic treatment of the cancer patient. So this remedy, ultra-high diluted RNA molecule, developed using clinically proven high technology to reduce the expression of ABCD1 genes to help support the use of chemotherapy drugs by allowing them to enter and kill the cancer cell. Now, with a modern medical oncology, we can take up a project, directly give your chemotherapy drug, directly give chemotherapy drug with our RNA molecule. How best we could able to protect or improve the chemotherapy. See, any doctor who is working, they can directly discuss to modern medical oncologists. Definitely, they will come forward to design a lot of experiments with these drugs because they know the sequence of RNA. So they do not know our classical homeopathic drugs, but they know the Human Genome Project. Next. So now that is first molecule. Then next coming to this, the second molecule, that is MENA and IN. This remedy should be administered to all cancer patients. The gene MENA is also known as ENHA, encodes a protein involved in regulating the assembly of actin filament. It also modulates the cell adhesion and cellular motility. It has been found that chemotherapy actually promotes the tumor spread or metastasis via one of the alternative transcripts of a transcript of mRNA called ABNC. How critically, when certain chemotherapy drugs are used, rather for arresting it promotes the metastasis. Another one of the big nightmare for the modern medical oncologist is this particular thing. So this can how we can address with our homeopathic RNA. So as might be expected, therefore, suppression of the expression of this particular gene stops the tumor spread. See, they have identified one particular gene. If it is overexpressed, that there is a fast spread of cancer. If you pacify, if you just slow down that process, the metastasis can be slowed down. So the remedy is this Onco-Metacare. We have just named metastasis prevention, Metacare like that, has been developed using clinically proven high density. Again, see, there is it has got from the patent road. Reduce the formation of MENA transcript to reduce the likelihood of chemotherapy-induced tumor. See, all the details in detail are available in the link given below. One can read very deeply into the subject for designing the experiment. Next. Now, another third and fourth molecules. This OncoCare 3, we have named it them. So, because these two molecules have a similar function, that's why I've clubbed together. Each of these two remedies should be administered to all the cancer patients. Formation and progression of the cancer cells is driven by an uncontrolled expression of the genes that promote the cellular proliferation. The cancer increases because of this uncontrolled expression of the cancer genes. So such genes, we call them as the proto-oncogenes. They encode the proteins that initiate the cell cycle. When the control control of the expression of these proto-oncogenes is lost, then they are referred as the oncogenes. Proto-oncogenes, everybody carries. When they become oncogenes, the cancer progresses. And progression of tumors. Uncontrolled expression of oncogenes, that is MYC and FOS, are a feature of many cancer cells. So therefore, the remedies, these 3 and 4, have been developed to target these MYC and FOS oncogenes to slow down the proliferation of cancer cells and support the benefits of conventional treatments. The constituents of these remedies 3 and 4 have been taken directly from another patent, which is available there. You can also go through it. Next. Now coming to the 5th molecule. This remedy should be administered to all the patients with germline or somatic mutation. Here, let me share something about this P53 gene. See, you have all experienced what is P53 is quarterback of a cancer immunology or quarterback of a cancer whole biology. This is like a king. If it is wonderfully there, nothing is going to happen. If it is got out of order, people will get cancer. So this P53 is also will be there in mutated form. They say that if we analyze the individual's gene status of this P53, whether it is natural or whether it is in a mutated form, all the mutated form of individual cancer patients will develop negative to the cancer. That means cancer will increase. Whether they may help in P53 classical cases, but in a mutated form, rather than benefiting, it does a lot of damage. And actually, even this again said by the medical oncologist. See, before going to the chemotherapy, one has to undergo, this is the simplest P53 mutation. If you are found to have a mutated gene, don't go for chemotherapy. See, that is the advice everybody gives, and we should also give it. But why these things are being not done, I do not understand. The gene P53 encodes a protein P53 that has a range of important anti-cancer properties, as I've already shared. It instructs the genetically damaged cells, such as cancer cells, to kill themselves, see, apoptosis. It also stops the cancer cells from growing and spreading also, as I've already shared. Many cancer cells are associated with a mutant form of P53. See, the mutated P53 gene, people are very negatively respond to the cancer, aggressively respond to the cancer. Rather, cancer will increase rather than decrease. Mutant P53 molecules encoded by the mutant P53 genes are unable to eradicate the cancer cells. They cannot get rid of the cancer cells. Next. So, more importantly, mutated P53 molecule promotes the tumor progression in the presence of chemotherapy drugs. See how bad we are doing it. Even see, mutated P53 individuals, when they are subjected to the chemotherapy and radiation, they get lot of nonsense, lot of aggravation in tumor. That's why we understand, instead of subjecting them for these things, they come out with, when you take the PET scan, instead of so much of drugs they are increasing, the reason could be this. See, if this is subjected, out of 70% of the cancer treatments nowadays which are going, they do not require any chemotherapy and radiation. But that's not being taken care of. So it has been discovered that persistence of mutant forms of P53 is supported by the protein HSP. Our RNA molecule is going to save the individuals from this. So therefore, the remedy Oncomutacare has been developed using clinically proven high dilution technology. Again, patent, you can go through this. Next. So another molecule, I am coming to it. There are total 9 molecules which we are working. So this is Oncorad 6, Adam 17. This remedy should be administered to all cancer patients, especially those who receive radiation therapy. See how beauty, now one can, how wonderfully we can use radiotherapy, one has to decide with the knowledge of this. The gene ADAM17, that is a disintegration metalloproteinase enzyme, ADAM, encodes for a zinc-dependent enzyme. Its role is to release a range of growth factors involved in the cellular proliferation. So recently, scientists have discovered that this ADAM17 gene is overexpressed in cancer cells. See, if this is overexpressed, again the negative response to the radiation therapy. So they have also shown that overexpression of the ADAM17 is associated with the tumor spread and poor prognosis. See, hats off to Human Genome Project. It's throwing a lot of light for diagnosis of the diseases, for even designing therapeutic applications. So importantly, from a treatment standpoint, overexpression of the ADAM17 renders the cancer cells insensitive to the radiation therapy. So that's why this remedy has been developed to increase the beneficial effects of radiotherapy by reducing the expression of ADAM. See, suppose being a homeopathic doctor, if you go and talk to your radiation therapist about this ADAM17, definitely he will curiously looking into it, curiously wants to experiment with our own RNA molecules also. Next. So now let me coming to this another 7th molecule, RNA remedy. So this remedy should be administered to all breast cancer patients. See this. The gap junction beta protein, that is GJB2, also known as the connexin 26, that is what they are called as CX26, is a protein that in humans is encoded by the GB52 gene. It plays an important role in control of cell or the cell interactions via the gap junction. See, when cancer means we are thinking only cancer cells, but even if you kill the cancer cells, cancer will not go. There are the cancer stem cells. You have to eradicate the cancer from the cancer stem cells. Stem cells cannot be left freely from our body. The cancer stem cells play an important role in the tumor progression and the recurrence of post-treatment. See, all the cancer cells, you have killed it. Then why it is recurred? There are cancer stem cells again which are producing cancer. Unfortunately, scientists have shown that this connection 26 is elevated in the cancer stem cells, and that is the necessary and sufficient for their maintenance. See, this particular gene, this protein is overexpressed in cancer stem cells. That is enough for becoming them into the cancer. If you speak all these things to a modern medical oncologist or radiologist, definitely they will look into our drugs, and definitely you can become a co-investigator with in the big cancer hospitals like this. My intention is only to just refresh you and make you aware of these things which are available in homeopathy. Next. So now the clinical observations also, the same continuation of 7th molecule, demonstrate a strong correlation between the increased CH26 expression in the breast cancer cell and decreased overall survival. See, when it is more expressed, the survival rate is very less in breast cancer patients. Moreover, expression has been shown to be associated with increased lymphatic vessel metastasis. See, this has got relation with metastasis also, increased tumor size also. See, if suppose in a cancer individual, if you just subject the individual for this CX26, maybe for other ovarian cancer also, but particularly in the breast cancer patients, you can immediately check that the size of the tumor, prognosis, why it is pure. You can throw a lot of light the poor response of the treatment in your breast cancer patient. This remedy has been developed to help and enhance the effectiveness of modern medical treatment as well as checking the breast cancer. Again, the details, you go to the patent expired. Next. Now coming to the last but one, another one is OncoCare 8 we put it. So these remedies, there is one PD1 and PDL1. How beautifully they are associated with protection and development of cancer. You will wonderfully appreciate, I think. So these I will share. So these remedies are to be administered to all the cancer patients to support the cancer cell-specific T-cell cytotoxicity. See, we were talking about a lot of radiation, chemotherapy in one angle, but our own immune system has got these cytotoxic T-cell which directly goes and kills the cancer cell. See, if this is wonderfully developed, who will prefer other therapies which have a lot of side effects and which have a lot of negative effects? So then why this particular thing is not working in our body also, I will throw a little bit light on you. The immune system plays a very important role in eradication of cancer cells, mainly by production of cancer cell specific cytotoxic T cells. T4, T8, T4, CD4, CD8 cells associated with immunological disorders, but for cancer, cytotoxic T cells. Even though A patient's immune system produces cancer cell-specific cytotoxic T cells. They are often failed to eradicate their cancer cells. Why? That is because cancer cells produce an abundance of cell surface protein called programmed cell death protein 1 ligand, that's PD1L. This is on the cancer cell. There is another marker, excess of PDIL on the cancer cells prevents the cytotoxic cell-specific T cells from killing them. If the cancer cell is overexpressed with the PDL1 receptors, our T cells cannot act on them. Why? Cytotoxic T cells also express a receptor for which PD1, this is programmed cell death protein PD1. One is PD1L. One is on the cancer cell, another is on the our cytotoxic cell. These creating, I will tell you. So, if these two are there, our efficacy of our immune system in killing the cancer cells is reduced. The binding of the PD1 on the cancer cell, specific cytotoxic cell to its receptor, and PDL1 on the cancer cell inhibits the cytotoxic effects of the T cells. See, these two are antagonists. Like. So the RNA-based remedies, OncoCare 8 and 9, we have clubbed together, have been developed to reduce the expression of PD1 and PDL1 on cancer cells and specific cytotoxic. PD1 should be less expressed on cancer cells. In a similar fashion, PDL1 should be less expressed in the T cells respectively. See, that is with a precise aim, we are planning these remedies out of the lot of research done, specific research done on cancer. Now it is our duty, if an homeopathy believes it or not believes a different way, but they can subject these all 6 molecules like any other classical homeopathic drugs on cancer, to a cancer people, then the things will come out. So importantly, this OncoCare 8, which targets the PD1, must not be administered to such a T cell leukemia cell. See, T cell also. How is also okay. Next, and then now let me finally come to the final molecule that we named as OncoCare 9, PARP1 gene. This OncoCare 9, it is to be administered to all the breast cancer patients with BRCA1 and BRCA BRCA1 and 2 mutations. Cancers such as the breast or ovary, which arise due to the BRCA1 and 2 mutation, and others develop a unique cancer-specific DNA repair pathway that relies heavily on the overexpression of a poly-ADP ribose, that is polymerase, that is PRP, shortly called the type. So PARP1 activity allows the cancer cell to repair their damaged DNA very efficiently, enabling them to continue dividing in spite of the application of conventional DNA damaging chemotherapeutic or the radiotherapeutic treatments. Thus, OncoCare 9 is to be used to support the benefits of the application of conventional treatments, including radiotherapy and chemotherapy. See, we are not telling the modern oncologists to take away your chemotherapy and or radiotherapy, which are the hallmarks of nowadays treatment. Rather, we are supporting their treatments for further effectiveness. Definitely, they will become our good friends, and they will also wonderfully look into these molecules. This is actually my opinion. And then I will not very deeply go into this thing. A few of the cases treated, I will not share much detail, but only we have treated a case of thymoma, post-operative post-recurrence since 2015. There are hiccups, I will let you know later on. A cause of breast carcinoma, adenocarcinoma, chest wall. These are all advanced cancer cases. And colon carcinoma, cervix, PRT status, and a sigmoid colon carcinoma. And actually, this I will only share one case. A 53-year-old male diagnosed with thymoma in 2015, underwent thymectomy, received concurrent chemotherapy, radiation therapy, and radiation therapy completed in 2015 itself. The whole body PET on 2018, recurrence with pericardial effusion. Cycle 1 palliative chemotherapy with a cap from 7-12-17. recurrent pericardial effusion. Pericardial centesis was done on 18. Balloon pericardiectomy was done on 18. Recurrent malignant pericardial effusion. Pericardial synthesis is being done on again in 2008. Earlier he was put on DNA molecules. There is no improvement. We have specially prepared for this particular a big industrialist. He got prepared himself all these molecules out of his own expenditure, and we are just now pulling on them. And so with this, I could able to complete my presentation. I might have taken more time. I do not know with the organization. And this is our RRA Gudivada. Of course, I am away from the council for the last 22 days. I got superannuated from the council on 31st January. And this is our research center at RRH Gudivada. Then you all know, thanks for your kind attention. And it's the nature's infinity and book of the secrecy. A little I can read and prostrate. So, sir, with this, I could complete my presentation. Sir, over to the organizers, sir. Okay, sir. Unmute.
Hello. Thank you, Dr. Raveender. So, Dr. Dahad, you please kick over and comment upon Dr. Raveendra's lecture.
Yes, sir, sure.
Next time we'll read the questions. If any question you see in the chat box, you can read
sure.
and discuss.
First, we should go through the questions. If anybody is having questions, then accordingly we can go ahead.
Yeah, please take care.
Hello. Hello. Hello, Dr. Raveender.
Sir, hello, tell me.
Good evening, sir. Actually,
Yes,
through the
evening,
chat
sir.
box, when I went through it, few questions are there. First of all, we are going through that, and accordingly, then we can go ahead. The
Okay.
first question has been asked actually where we can get these remedies, and is it easily available? Second was related with the posology and general guidelines regarding the administration of it, etc. So can you enlighten in the brief?
Okay. Sir, actually, the sequences we have already standardized. These sequences we have to get isolated from a high technical genetic laboratory. RNA sequence, required RNA sequence, we will get. There are some identified organizations are there in India. We are getting isolation from the Delhi technologies like that, life technologies, and there are some other organizations also. So that isolated RNA, there is a procedure how to prepare 6 potency in homeopathy as per to the certain basic properties of the RNA, and that once we got isolated these molecules, we can prepare in any homeopathic laboratory or any homeopathic pharmacy. We can prepare 6 potencies. So there is a procedure for it, and for the viewers benefit, we are shortly going to start a course on these things, including a basic course on cancer. Hope within a month we are going to float it on the net. So that in that course you are going to learn lot of cancer, lot of immunology, and this is headed by one of an international scientist from UK. He will be answering all your questions, and he will be designing your papers, and he will be you need not worry it. He will try to give consultation to your cancer cases also, even while you learning these things. So that actually I would have shared. So hope we will share you with the Dahad. We will share you when that course comes into existence. So that course we are planning to teach you basically the basic immunology, basics of cancer, and the basic treatments of cancer. What are the problems with the modern medical treatments? How best we are planning these things, and how best you can counsel a cancer patient also. How best you can advise a cancer patient to go for. So all these things we are going to incorporate in our cancer code. Hope shortly, in less than 1 month, we are going to float it on the net.
Yes, sir. Thank you. Any more question from the participants? You can unmute yourself and ask the question, or just one more. Chinta.
Yes.
Hello.
Hello.
Availability of the medicine.
The availability of the medicine, we have to prepare on our. If somebody really wants, we can supply. See,
Okay,
this is actually only to doctor we will supply. Okay.
Yeah. Sure.
Okay.
Let me check the chat box. Actually, they are asking.
They can mail to my address also. Enquiries they can make. I will try to address them. Okay.
Yeah. Please just once
Any
you show
questions
your
related to availability or whatever it may be, about course and all the things, I will try to respond.
You email and mobile number. Can you please?
Okay. And okay, sir. Link we will provide you very shortly, and all the things. Yes, they're there for breast cancer. I am looking at these questions. Sir, I will try to definitely address them. We will send the email to the hall. And I think my PowerPoint is, Dr. Dahad, you may share my PPT to all if they need, to the participants. Okay, you can keep it. See the result of treatment. We have started very recently, and one can go for. Okay, one can go for a lot of research on these things. This okay. We have to first learn from the course sequence. We have to got it from any genetic laboratory. Okay, so my you can pose. I have provided my email ID and my mobile number also. Okay, in chat box. Sir, giving the medicines, is there any specific site?
Yes, sir. Thank you very much. Dr. Muktinder Singh.
We have to get
any more comment or should I conclude?
That I will provide you. Not a big matter. Medicine ourselves we can make. Yes. How to make medicines ourselves? It's a procedure. I will share. Okay.
Yes, sir.
Because based on the RNA and DNA properties, they have designed one method. And how to make medicines ourselves. Okay, fine. Yes, sir. So, Daji, I have almost answered the questions in the chat box. And thank you, sir, very much. One can get a lot of innovative ideas out of this because this is done by a wonderful scientist from the other side. So that's why we could able to make all these things. So if any organization can plan a lot of research with these things, we can provide. If they want in vitro studies, they can go. In vivo studies, animal models also they can go. But for all the things that are there, because sir, Daji, you may share my PowerPoint.
Hello, Dr. Muktinder Singh.
Sir, are you hearing me? Actually, my charging gone out of order, I think. Am I audible to sir?
Yeah. Anyway, Dr. Muktinder Singh.
From my laptop as well as from the mobile, the charging is getting 0.
Hello, Dr. Raveender.
Hello, Dr. Dahad.
One second. Some technical issues.
Take minute.
Yeah. Now I am audible. Hello.
Dr. Dahad.
Dr. Muktinder Singh. Should I conclude the session?
Yes, I'm concluding it. So thank you, Dr. Dahad and Dr. Raveender. So I have an announcement that on 28th of February, we are planning a quiz session by Dr. Padma from Visakhapatnam. So details would be posted soon. Suppose you follow detail if you want to participate actively into the quiz. So detail will be given. So I thank on behalf of IIHP Dr. Raveender and Dr. Dahad sir and whole IIHP team, Padam Shri Dr. V.K. Gupta sir and Dr. M.A. Rao sir. So with this we conclude the meeting. So hope we'll be meeting next time, and Dr. Raveender's insight into the topic would definitely help the young fraternity and all the IIHP fraternity. Thank you once again.
Thank you very much, sir. Thank you to all. Thank you.
Thanks a lot.
Thank you.